Hi, I'm Ariana, a medical intervention student. Here are 5 of the most challenging topics on the BACE exam. Monoclonal antibody production. It's important to understand that monoclonal antibodies are proteins made in a lab that bind to one specific antigen. The procedure can be condensed into 5 steps. Collect immune cells from the animal, lymphocyte fusion between B cells and myeloma cells, optimize and screen hybridomas producing the correct antibody, cultivate and expand the hybridomas, and extract the monoclonal antibodies. I recommend being well versed in the adaptive immune response by learning the function of B cells and T cells. Visit the BioRad Life Science tutorial videos for help. Protein analysis. There are 4 different protein analysis assays for the BACE exam. Gel electrophoresis separates and analyzes DNA and RNA by size and charge. SDS-PAGE is similar to electrophoresis but focuses on proteins and requires the SDS molecule to neutralize their charge. The Western blot test identifies specific proteins in a mixture with antibody markers, and Bradford assays measure concentration of proteins in solution. I recommend visiting the Khan Academy Biotechnology Unit or the Biotility website to remember the differences between each of the assays. Different microscopy techniques. The main type is a light microscope, which uses visible light to illuminate a specimen, such as a bacteria cell. The subtypes of light microscopes include bright field microscopes, phase contrast, and fluorescence microscopes. Electron microscopes use a beam of electrons instead of photons to image the specimen and gain information on its structure and composition, while confocal microscopes use laser imaging. I recommend looking at the Amgen Introduction to Biotechnology website for more information. There are 4 types of ELISA testing: direct, indirect, sandwich, and competitive. The BACE exam primarily focuses on direct and indirect ELISA. Direct ELISA, the primary antibody binds directly to the antigen-coded plate, while in indirect ELISA, the primary antibody is followed by a secondary enzyme-linked antibody. I recommend watching digital animations of both procedures and then testing yourself by drawing the process from memory. In sandwich ELISA, there are 3 antibodies involved, creating the sandwich structure. Polymerase chain reaction is a laboratory technique that amplifies a small amount of DNA or RNA. PCR uses a thermocycler to denature DNA strands, anneal them with primers, and then extend them. It's important to note that this cycle is repeated about 25 to 40 times to amplify a single sequence at about 72 degrees Celsius. For more information, visit the National Center for Biotechnology Information.